I also just want to start by thanking Simona and Francesca, congratulating you on the success and bringing all these people in from around the world. It's amazing what you're doing, and it's very impactful. So congratulations and thank you. I was going to shout out that very recently the Gould Syndrome Foundation has had a reorganization, new leadership, and one of the members here, Tim, is here, who's the treasurer. But we've touched on that a little bit, so in the interest of time, I'll just keep moving. These are the members of my lab right now. I always like to start and acknowledge them and thank them because they're the ones that are actually doing the work that I get to stand up here and present. And these are the people who we're working for. So we're working for the patients, and we're working for the families. We're trying to find ways that we can impact this. So. My lab works on collagen for alpha-1 and collagen for alpha-2. We've been doing this for about 20 years now, and essentially our goal is understand what they do. So these are fundamental proteins that very little is known about their function, and understand when they're mutated, what's going wrong. So essentially, what is the disease mechanism, such that we can then exploit that and capitalize on it and develop mechanism-based interventions. So that's what I'll talk about today, but of course at the end I'll also talk about mechanism-agnostic interventions, so things that might be coming down the line even if we don't know the disease mechanisms. So this is just a brief overview of what I'm going to talk about, tell you a little bit about what type 4 collagens are, a little bit about what they do, and then some potential mechanism-based or mechanism-agnostic interventions. So the collagen superfamily is it's one of the major extracellular matrix proteins. There's 28 different kinds of collagen in your body. And we're talking about type 4 collagen. Despite being called type 4, it's actually the ancestral collagen. So what makes a collagen is that the protein has a repeat of glycine XY, GlyXY, GlyXY. If you're a protein and you have a GlyXY repeat, you're a collagen. And those GlyXY repeats form what's called a triple helix, okay? And so around 750 million years ago, when single-celled organisms started sticking together and becoming multicellular organisms, they started making type 4 collagen. So it's been around for that long. And it's present in every tissue in every animal of the animal kingdom. So these are really prevalent. And yet again, we really don't know much about what they do. So there are 6 different genes that make type 4 collagens. Collagen for alpha-1, collagen for alpha-2, and then collagen for alpha-3, 4, 5, and 6. Collagen for alpha-3, 4, 5, and 6 have much more restricted spatial distributions. So for example, collagen for alpha-3 and alpha-4 are primarily located in the glomerular basement membrane of the kidney, also in the cochlea and in places in the eye. And so it has been known since around the 90s that mutations in collagen for alpha-3 or collagen for alpha-4, collagen for alpha-5 cause Alport syndrome, which is kind of a sister disease. We can learn a lot from that field. And so what they all share is this same protein structure. So I don't know if this, yeah, I don't know if you can see that, but at the carboxy terminal end of the protein is the NC1 domain. That just stands for non-collagenous domain. And so the NC1 domain kind of forms into a ball. And then most of the protein, though, this long strand, is this repeat of glycine XY, GlyXY, GlyXY, GlyXY. And then at the amino terminus is this a small domain called the 7S domain. And I'll tell you a little bit about those things in a moment. The NC1 domains of collagen for alpha-1, alpha-2, 3, 4, 5, and 6 are responsible for recognizing the binding partner and initiating assembly of the heterotrimer. And so despite the fact that these 6 proteins in theory could form 56 different combinations of homo or heterotrimers, in reality, the only ones that have ever been identified in tissues are these 3 heterotrimers, collagen for alpha-1, 1-1-2, meaning there's 2 alpha-1s and 1 alpha-2, 3-4-5, and then 5-5-6. And what happens when they're being made by your cells is that they're co-translationally translocated into the endoplasmic reticulum. Okay, this is a bit of jargon, but that means the proteins are made into this compartment where they're then going to assemble. When they're in the endoplasmic reticulum, those globular NC1 domains find each other, right? So the globular domain of collagen 4 alpha 2 finds 2 domains of collagen 4 alpha 1, and then they start winding up like the braiding of a rope. They undergo some modifications that won't be important for today's talk, but that protomer, it's called, this heterotrimer of 2 alpha 1 and 1 alpha 2, gets secreted into the basement membrane, and then it forms a network, like a fish net. So these globular domains form head-to-head interactions, and then at the other end, those 7S domains form anti-parallel lateral interactions, okay? And so in that way, it stitches itself together and forms this, like a fishnet, a flexible sheet. Now, what happens, these mutations are dominant, right? So you need 1 mutant copy. And so if you imagine being inside this compartment of the cell, assuming that these proteins are all randomly assorting, a quarter of the time, 25% of the time, collagen 4 alpha 2 is going to find 2 normal copies of collagen 4 alpha 1, and those ought to form a normal heterotrimer and be secreted. And some of our early data showed that a quarter of the time, collagen 4 alpha 2 is going to find 2 mutant alpha 1s, and these don't get out of the cell. The cell quality control mechanisms recognize this, and they stop secretion of those. And then 50% of the time, so the largest proportion of trimers will find 1 normal alpha 1 and 1 mutant alpha 1, and we didn't really know what the fate of those were. But essentially, this sets up 3 potential disease mechanisms. You could have a toxic effect of this intracellular protein, right? The cell can choke on this accumulated protein. Alternatively, you could have an extracellular deficiency. If the cell is impairing secretion, or the mutant proteins aren't being secreted, you don't have enough in the basement membrane. A third possibility is that this proportion of heterotrimers is actually being secreted, and that the presence of the mutant protein in the basement membrane could be damaging. So that's what they are. What do they do? I told you that they've been around for about 750 million years, and they're throughout the entire animal kingdom. But the truth is, we don't really know what they do. They could do many things, right? They bind to cell receptors. They bind to signaling receptors, and so, you know, we don't really know what they do, but we know what happens when they're not doing it, right? When you have mutations in collagen 4 alpha 1 or collagen 4 alpha 2, you get this multi-system disorder. You know, we've all talked about some of the organs that are typically understood to be involved, but the truth is that we've seen pathology in every single organ that we've looked at carefully. Sometimes that means using transmission electron microscopy, but it really does affect, or can affect, every organ. And this is just a quick slide to show you. I mentioned we work in mouse models, and they very well replicate the human disease. So what do they do, and can we develop mechanism-based interventions? So one of the things that we discovered was that type 4 collagens seem to control TGF-beta signaling pathway. And so we had 2 manuscripts, one looking at the cerebral vasculature and one looking at ocular development. And essentially, the short version of the story is that this is data from the cerebral vasculature. Genes that are controlled by the TGF-beta signaling pathway are upregulated in the blood vessels of the brain of COL4A1 mutant mice. And so that's just shown. All these red dots are from the mutant mice, and then the black dots are control mice. You see significant elevation of these genes. And then shown here is an assay that we do to sort of measure the severity of intracerebral hemorrhage. We use this stain, and the blue indicates a place where there's been an intracerebral hemorrhage. And what this shows is that if we genetically reduce TGF-beta signaling, so we cross a collagen for alpha-1 mutant mouse to a mouse that's missing one copy of TGF-beta1, it actually significantly reduced the severity of intracerebral hemorrhage. So that suggests that one of the functions of collagen for alpha-1 and collagen for alpha-2 is to suppress TGF-beta signaling. And when you don't have enough collagen, you get too much TGF-beta signaling, and that suppressing TGF-beta signaling was beneficial in this regard. However, there's limitations to trying to target these, what we call the distal targets. It's possible that type 4 collagens have 5 functions, or they could have 50 functions, or they could have 500 functions. So being too specific about manipulating a distal target might fix one thing, but 49 other things still aren't functioning properly. And so we hope to then find something that's a little bit more proximal. Now, the story that I'm going to tell you is capitalizing on the variability, right? Everyone talks about how there's a broad phenotypic spectrum. So we wanted to understand what are the things that determine whether a particular mutation has severe outcomes or milder outcomes. And what are the things that determine where you might fall on that spectrum? This is an old slide. There's 50 or so mutations shown on this slide. Today there's over 400 or so mutations that have been reported, but the story is the same, is that they tend to affect these glycine residues throughout the triple helix forming domain. And so the question that we wanted to ask is, does the mutation determine or influence where you fall on the severity spectrum? And so this is called allelic heterogeneity. Do all mutations behave the same way, or is there allelic heterogeneity? And so to ask this question, we've been studying an allelic series of mice. So each one of these shows a mutation that's in an independent strain of mice. This one down here is S1582. So this is a mutation that's in that NC1 domain, the globular domain. The rest of the mutations are all in the triple helix forming domains. All but one of these is a glycine missense mutation. The only exception is this one. It's a deletion of an exon, but importantly that it maintains the frame. So it essentially skips 17 amino acids, but the rest of the protein is translated in frame. So we took each of these mutations, we crossed them onto a uniform genetic strand of mice so that every mouse is genetically identical, except some of them have one mutation, some of them have another mutation. And then we took cells from those mice and looked at the efficiency of collagen secretion, essentially. So in the top panels, on the left side is a fibroblast from a wild-type animal, and collagen for alpha-1 is labeled in red. And then in green is a protein that's involved in collagen biosynthesis. And that protein stays within the endoplasmic reticulum. And then on the bottom there is the merge. So the yellow labeling shows all the collagen that's still in the endoplasmic reticulum. And then you can see on these arms, there's some red collagen that's getting out of the endoplasmic reticulum. So that's being secreted. So then if you jump over to the right side, this is the deletion of exon 41. And you can see that all the collagen is inside the ER. There's not very much that's getting out. It's mostly not being secreted. But in contrast, if you look in the middle panels, this is this mutation down here, G394V. It looks a lot more like the wild-type cell. There's plenty of collagen that's being secreted there. So we wanted to quantify this. We took all of those cells, and we did a Western blot to look at the levels of intracellular collagen. And what we found is this interesting position effect. So each of these is a different mutation, and what's shown on this graph is how much collagen there is inside the cell. I'll come back to that. I know I went quickly. So then the second thing that we did was take each of those strains of mice, we aged them out to 8 months of age, and then we serially sectioned the brain, stained it with this stain to see where there's hemorrhage, and then quantified the amount of hemorrhage in one strain versus another strain. And this is the graph that we got across the bottom. So if you compare that to the intracellular accumulation, we start to see a correlation. And what's happening, there's potentially 3 different genotype-phenotype correlations. First, this domain effect. So this mutation that's in the NC1 domain doesn't really have very much intracellular accumulation and had almost no intracerebral hemorrhage. Then mutations that are within the triple helix forming domain had this position effect. So the mutations that are nearer the carboxy terminus, nearer the NC1 domain, tended to have more intracellular accumulation and tended to have more intracerebral hemorrhage. Mutations that are nearer the 7S domain, nearer the amino terminus, tended to have less intracellular accumulation, tended to have less intracerebral hemorrhage. Finally, there's also this allele-specific effect. We don't really know what's going on, but I'll point it out. The deletion of exon 41 respects its position in terms of intracellular accumulation, but it's disproportionately severe. And we don't really know what's going on there yet, but we're working on it. So the general rule that we took home from this is that impaired secretion is important, right? So if it's true that this population of heterotrimers always gets out and this population of heterotrimers never gets out, then the difference between a severe mutation and a mild mutation is the fate of this population of heterotrimers. In a severe case, they tend to not be secreted, and in the milder cases, they tend to be secreted. So then we said, okay, well, if we can alter the fate of that population of heterotrimers, then maybe we can take an animal that was going to have a severe disease and instead, make it better. So this is what we're calling this mechanism-based proximal target. So this is Genki and Mao. What they did was treat mice with something that's called a chemical chaperone. It's a small molecule that has protein folding properties. It helps proteins that are misfolded to be folded and secreted. And in these panels are both mutant mice, one that didn't get treated with the 4-PBA. And the green is showing you the blood vessel. This is a retinal blood vessel. And the red is showing you collagen. And this collagen's all stuck inside the cells. And then the mouse that was treated with the chemical chaperone, you see the collagen is actually now labeling the basement membrane of that blood vessel. And then when we treated the mice, this signal that you're seeing here is TGF-beta signaling, right? So this is a mutant animal and it's wild type littermate, and this is a mutant animal that was treated with 4-PBA. So this drug promotes the collagen secretion, it reduces the TGF-beta signaling, and sure enough, if we quantify intracerebral hemorrhage with that stain, we see that the mice that were treated with the chemical chaperone had milder or less severe intracerebral hemorrhages. However, this is the cautionary tale. In those same mice that we aged to 8 months and quantified intracerebral hemorrhage, we also looked at a number of other phenotypes. This particular one is looking at the skeletal myopathy. So looking at skeletal muscle, and you look for non-peripheral nuclei, which are the blue dots. The blue dots should be sitting on this green ring, but in a damaged muscle, they're not. They move to the center of that circle, and sometimes there's multiple blue dots inside the circle. And so we quantify those, and what we found is shown on the right. Interestingly, the mutation that's in the NC1 domain, it still is very, very mild. There's really very little wrong with these animals. And the deletion of exon 41 is still disproportionately severe for some reason. But the big shocker was this mutation, G394V, right? If you remember, that mutation had very little intracellular accumulation, had very little intracerebral hemorrhage, but it had the most severe skeletal myopathy, right? So this protein's getting out, but it's causing the more severe myopathy. So there's some sort of subdomain effect, we think, okay? If you notice on this cartoon, that mutation is sitting near this green stripe that I didn't tell you about. That green stripe is an integrin binding domain. Integrins are cell surface receptors for extracellular matrix proteins, including collagens. So what we think is happening here is this mutation is actually getting out of the cell, but it's not binding to its integrin. It can't. So what happens then if you treat those mice with 4-phenylbutyric acid, the chemical chaperone, it actually makes the muscle phenotype even worse. You're getting more of that mutant protein out there, but it still can't do its job, and so it exacerbates this part of the phenotype. So this is just quantification. The first gray bars are a mutant mouse, and then this is the mutant mice that were treated with 4-PBA, and they exacerbated that phenotype. For a different mutation, the deletion of exon 41, we give those mice 4-PBA, and it improves the skeletal myopathy, okay? So this is a limitation of trying to target that proximal insult, the protein folding and the impaired secretion. We don't know, I'll make up a number. Maybe 85% of patients will benefit from a chemical chaperone therapy, but 15% you might be exacerbating things. The problem is we don't know where those functional subdomains are on the protein because we don't know all of collagen's functions, right? We don't know the functions, so we can't possibly know where the functional subdomains, so we can't genetically stratify patients that have a mutation in a subdomain that's going to be harmful to get that protein secreted. So where does that leave us? Those are the two examples of mechanism-based interventions. What about mechanism-agnostic interventions? So this is just a fancy word for gene therapy, right? So what about gene therapy? This is what everybody wants to talk about. Can we go even further back into this pathway to the DNA or to the RNA? So there's a lot of open questions that need to be answered before you can undertake these kinds of approaches. When are you going to treat? When can you treat? Okay. Collagens are notoriously stable proteins. There's type 1 collagen, these are fibrillar collagens, that they estimate the half-life is about 120 years. Okay, so you might die with the collagen that you were born with. In one study in mice suggested that type 4 collagen is produced a lot during embryogenesis and then it drops off postnatally. And then in those mice, it looked like the vascular endothelial cells were making most of the collagen, right? But we need to know what's the turnover of collagen. That's going to determine what the therapeutic window is. If the collagen's no longer being made, it doesn't matter if you go and correct the gene because it's all been made and it's stable. So we need to know what the therapeutic window is. We need to know where. Which cells are we targeting? So if you're thinking about some sort of CRISPR editing, you usually have to target your cargo to the cells of interest. But which are the cells of interest, right? I just told you that a study said embryonically it's mostly the vascular endothelium that makes it. But in adults, it looks like it's the mural cells that make it. It's also possible that in a disease setting, there's other sources. It could be perivascular fibroblasts. So we need to know which cells we're trying to target, and those cells could change over time. And then finally, how? What kind of gene therapy are we talking about? Can we just add back more wild-type copy of COL4A1? Would that be good enough? Or is the mutant version still sort of having that dominant interfering effect? In which case, can we just do an allele-specific ablation? Can we knock out the mutant copy? Or is that going to be bad because now you don't have enough? Do we need to actually go in and correct the nucleotide? So to answer these key questions, we developed this new tool. It's collagen 4-alpha-1 that has a fluorescent tag on it. So this is so that we can see the protein in the mice. And it has some functionalities that will help us understand where and when the collagen is being produced. So at baseline, the collagen 4-alpha-1 is tagged with this M cherry right after the signal peptide. I didn't talk about this. This is 27 amino acids. It just gets cleaved and thrown away. So this is hanging off the end of the protein. Okay, so under baseline conditions, and the example I'm showing you on the right is a cerebral blood vessel. All of the basement membrane collagen should be labeled with M cherry. But then if you cross it with a mouse that carries CRE, right, it's going to switch it from M cherry to GFP. So now all the collagen in the basement membrane should be labeled with GFP. Now, because this is CRE dependent, we now can use all the sort of functionalities that CRE have. We can use a cell-specific CRE. So for example, on the bottom right, this is an example where we used, if you use a CRE that's expressed only in the vascular endothelium. Okay, so all the other collagen in the body should still be red, but anything that's made by the vascular endothelium should be green. Okay, in contrast, you can also use an inducible CRE. So this is a CRE that's expressed in every cell, but it's not active until you give a drug. So in this example, all of the collagen that's produced before you give the drug is going to be red, and all the collagen that's produced after you give the drug is going to be green. And then finally, with a different recombinase, with flip recombinase, it's going to truncate the protein right after that M cherry. And then we put a nuclear localization tag on it. So what should happen here is you won't have any collagen labeling in the basement membrane, but the M cherry should stay inside the cell that's making it. So it'll allow us to identify which cells are making it. So just the proof of concept of this, we're looking at the lens capsule because it's a really thick, prominent basement membrane that only has one source, the lens epithelium. So this is a mouse that doesn't carry the tagged allele. This is a mouse that carries the tagged allele at baseline. And then when we cross it to a ubiquitous Cree, you can see that there's no more red collagen. It's all green collagen, and it's really quite beautiful labeling. So then we wanted to do the inducible Cree, right? So we're going to give a drug that turns the collagen, all the collagen that's produced from that moment on should be green. And so Mao gave the drug, tamoxifen, at postnatal day 1, 2, and 3, so the day after birth. She gave it 3 days in a row, and then she looked at the lens at day 7. And what you can see is that there's still some red collagen there, but it's a very thin line. So this is all of the collagen that was produced before birth, and then this is all of the collagen that was produced after birth. And you can see the stuff that was newly produced is sitting beside those lens epithelial cells. But of course, the lens isn't what our most major interest is. It's the cerebral blood vessels. So she did the exact same experiment looking at cerebral vasculature. So she gave the drug postnatal day 1, 2, and 3, and then looked at the blood vessels at day 7. And this is the control. This is the mouse that didn't have Cree. This is the mouse that did. So at day 7, there's still a little bit of red collagen, but most of it's green. So that suggests in this first week after birth, there's still a lot of ongoing new collagen deposition. This is good, right? Because we want a postnatal therapeutic window. However, when she did the same experiment in a mouse that was around 6 weeks of age, about a month and a half, after 1 week, there's still a lot of red collagen, and there's barely any detectable green collagen. Then if she gives it another week, waits 2 weeks, you're starting to see a little bit more green collagen. But this is after 2 weeks. So this is telling us that the collagen production is decreasing with age. And so the therapeutic window, time matters, and earlier is better, essentially. Where I told you that the source of the collagen could change over time. Maybe embryonically it's mostly endothelial, maybe in adults it's mostly mural cells. And so using this recombinase, this flip recombinase, we can identify which cells are making it. So again, I'm just showing you the proof of concept in the lens capsule. This is the control, and then this is a mouse that's been crossed with flip, and you can see all the collagens within the cell. This is just labeling a membrane to show you that that red signal wasn't just sitting on top of the cells. So Xiaowei did this experiment where she crossed the tagged mouse to a flip mouse, and this is what she sees. And this is all very preliminary. But what you can see is that it's not labeling everywhere. It's not labeling every blood vessel, let's say. So at this moment in time, this mouse is about 6 weeks old. There's only some cells that are actively producing it, or producing it at a level that's detectable, right? And for the real aficionados, I mean, it looks to me like penetrating arterioles might be the vessels that are producing the most collagen at this age. So the next steps are to capitalize on this intracellular M-cherry. So you make single cell dissociations, you flow sort for only the M-cherry cells, and then do RNA sequencing. So we can identify all of the cells that are making collagen at any moment in time. And we'll do this embryonically, postnatally, et cetera. And then the final question, how? As I mentioned, we can either try and express more wild-type collagen. We could maybe knock out the collagen. Do we need to do gene correction? The problem with gene correction is this N of 1 problem, okay? Collagen for alpha-1 has about 350 glycine residues, and collagen for alpha-2 has about 350 glycine residues. And there's about 1,400 unique DNA changes that could essentially lead to a similar problem. So if you're making a gene therapy that's going to go in and correct a nucleotide, you're going to need 1,400 independent correctors. That's not ideal, at least for now. So remember, I wanted to remind you that the mutation in this NC1 domain, it secreted pretty efficiently, and it really didn't have much of a phenotype. So what we think is happening here is that if you mutate that domain, it doesn't fold properly, and so the other NC1 domains don't recognize it. So essentially that protein's disappearing. So it's kind of like a null, like a loss of function. So that suggests that knocking out, if we can knock out the mutant allele, that might be beneficial. And I already showed you this. The first time I showed you, I was trying to show you the cells that are making collagen. But remember that this mouse has only one wild type copy. The other copy has been knocked out. And so these mice looked pretty normal. And so then we went through a number of different organs. So this is looking at the eyes. The first two are just controls. And this is a mouse that's only got one copy of collagen for alpha-1. The other copy has been knocked out. There's a little bit of abnormal iris vasculature, but it's fairly subtle. Histologically, the eyes look perfectly normal. I won't dwell on this. I'll just give you the punchline. We looked at the skeletal muscle. They don't have significantly elevated non-peripheral nuclei compared to their littermate controls. We looked at the kidneys. The kidneys look completely normal. We looked at the cerebral vasculature, okay? And 9 out of 12 mice had no evidence of any lesions. There was one mouse that looked like maybe the ventricles were enlarged, and there were 2 mice that had a small leak when we do an intravenous injection of fluorescent tracers to see if the blood-brain barrier is compromised. 2 animals had small leaks, but they're far, far, far less severe than animals that have glycine missense mutation. So it suggests to us that more of a pan-mutational approach, if we can knock out the mutant allele, that could be a beneficial approach. This opens up the possibility, at least, for, say, antisense oligotherapies, let's say. So things that just selectively knock down the mutant alleles. So to summarize, the general mechanisms are that you have these semi-dominant mutations, they impair secretion, and you get extracellular deficiency. So then the collagen's not getting out, it can't do its functions. One of those functions looks like it's TGF-beta repressor. So if you don't get the collagen out, now you have excess TGF-beta. We can possibly target that as a distal insult. We can also try and promote secretion. So this is a more proximal approach. But I think if we look 10 years down the road, where are we going to be, or where do we want to be? And it's going to be gene therapies. So there's still a number of those things to be worked out, but understanding the time and location and the types of approaches are going to be key. So just a quick acknowledgement to my funding sources, some of my key collaborators. And for anyone who's interested in collagen research, I'm actually the chair of this Collagen Gordon Conference. It's the preeminent collagen research, International Collagen Research Conference. So please come there if you'd like to learn more. And once again, just thank my lab members and thank you for your attention.
Thank you so much for this exciting presentation, and I think we are running out of time a little bit, but maybe at least some few questions.
Thank you very much for your talk. I'm completely aware that gene therapy or new drugs, new molecules are the future for these patients as in Alport syndromes. But meantime, I wonder, do you think that it would be room to treat this patient with RAS blockade drugs, which are able to block the synthesis of TGF-beta? Also without proteinuria or hypertension, for example, because I think that there are very similar points between this disease and Alport syndrome disease, and I think that now RAS blockade is even the main therapeutic line.
Yeah.
So I wonder if you are some ideas on it.
I have a lot of ideas about it. I mean, number one, I think that yes, we can try and learn from what they're doing with Alport, because they're, like I mentioned, I consider them sort of sister diseases. I also talk to a lot of patients, and I am very clear, like, I need to stay in my lane and not advise what may or may not work. I think, though, that, I think the major goal of those drugs is to reduce, are they not, to reduce pressure, right? And I think what's happening there, my understanding, is that they're, essentially the high pressure is pushing albumin across the barrier, and it's getting to the tubular cells, uptake the albumin, and it's toxic to the tubular cells. So I'm not sure that what it's accomplishing would translate here. That's not to say that they might not work for other reasons, but I don't know.
Thank you.
Thanks.
Any other question? Yeah, over there. There.
Well, hi. Very, very nice talk. Thank you very much. I have a question about the actual amino acid decomposition of the protein. And of course, glycine are very important. But if you look at the sequence, there are also a lot of proteins.
Yeah.
And I was wondering whether you can comment on why you don't really get mutations on proteins, although it seems that they can structure these polyprotein type 2 helices and may have a role. And if you can comment on this, thank you.
Yeah, I'll make a little bit more of an even broad comment. I think that there's lots of mutations that aren't in glycines, but we just don't know what to do with them. So, glycine mutations, we now can say that's a smoking gun. I'm confident that that's pathogenic. Anything that's not a glycine variant, we're kind of guessing. And now prolines, yeah, there's a lot of prolines because GPP, they do help to form that stable triple helix. So I'm sure there will be proline mutations, but yeah, it's hard. It's just hard to know right now. And some prolines get hydroxylated to either 4-hydroxyproline to stabilize the structure, or 3-hydroxyproline, which in other types of collagens has been shown to be important for extracellular functions. So I think that there's this entire class of mutations out there. Glycine in general, glycine missense mutations are going to impair secretion, but you can have mutations in other amino acids that won't affect secretion, but will affect the extracellular function. So, lysines, for example, lysines also get modified, and so there are pathogenic lysines that have been identified. So I think that's a whole class that we don't have a good handle on those at all.
Thank you.
Yeah.
There is a question over there.
Many thanks for this very nice presentation, Doug. I have a question. At the end of your talk, it's like if you were mentioning that haploinsufficiency in the mouse would not be that severe, that would allow to use therapeutic approach that would transform the patient to an haploinsufficient patient. But I think we need to have information because, patients with stop codon,
Yep.
some of them have severe phenotypes. Yep. As far as I remember, there's no stop codon in the UK Biobank, in the 450,000 people. Yep. And so it might be a little bit different between the mouse and the humans. I know that's just...
I completely agree. If you look in the UK Biobank, loss of function variants are underrepresented, which suggests that they can be pathogenic. They're being selected against. Yeah. That's true. That's true. And you're right. It could be species differences, human-mouse differences, but I think, I don't know that I can say more than that because what we're studying could just be in the mice. It does, I think, from what we know about it, it does make sense that the dominant interfering mutations will affect 75% of the heterotrimers, right? So if you only have one copy, the remaining 50% will all be normal. So you are presumably skewing it towards, better outcomes or more collagen being sufficiently secreted. So yeah, but I don't know what to do with that.
Yeah. It's a big problem when people want to use anti-nucleotide, antisense nucleotides. And this is very serious question.
Yeah, thanks for that point.
Okay. Hi. I have one curiosity. So in your haploinsufficiency model, do you have evidence of transcriptional changes, maybe also for COL4A2 or other relevant transcriptional changes that can explain you a little bit what you see? Or
yeah, we don't see transcriptional changes. We haven't looked more broadly than at COL4A1 and COL4A2, though. But we do, we have less COL4A1. COL4A2 doesn't seem to change. Yeah, I don't think we looked at any other genes. Yeah.